We're just going to now move on to our next speaker. So I'm delighted to invite Professor William Liu to speak, and he is the Deputy Director of the Chinese National Influenza Center. Professor Liu graduated from the School of Public Health at Peking University Health Science Center in 2005 and received his PhD degree from the Institute of Microbiology at the Chinese Academy of Sciences, and then received his postdoctoral training at the Yale School of Medicine. The main research focus of his group is the T-cell recognition of emerging and re-emerging viruses, such as SARS-CoV-2 and influenza viruses. He's published more than 120 papers in leading journals, including Nature, Immunity, PNAS, Journal of Immunology, and Journal of Virology. He's the Deputy Editor-in-Chief of Biosafety and Health, invited guest editor of Frontiers in Immunology and Science China Life Sciences, and also editor for Infectious Diseases and Immunity. Professor Liu has worked as a Chinese public health team leader in Africa during the Ebola epidemic. And during the COVID-19 pandemic, he worked in Wuhan in China for over 100 days. So we're very honored to have Professor Liu come to talk to us today, and we appreciate his time. And we're looking forward to hearing his talk entitled, Seeing the Antiviral T-Cell Immunity, Believing the Epitope-Based Vaccines. Thank you very much, Professor Liu. Over to you.
Thank you very much. It is my pleasure to be here to share our recent, researches and our opinions about the, the antiviral T-cell immunity and also the potential epitope-oriented vaccines. I'm William Liu from China CDC. Actually, my talk will talk about the flu and the COVID-19 pandemic a little bit. And also the some immunity studies on flu and COVID-19, and also we will show something about the structural studies of MHC1 moleculars. So actually, when we go to the COVID-19, actually we have to go back to the influenza pandemic. Actually, we can see from the about 100 years ago, from the very beginning, the Spanish influenza virus, actually have emerged in 1990. Actually, we can see, the swine viruses segments have been, confirmed that it can reassortant with the human influenza viruses. So actually, from that day, actually, for the H2N2 and the H3N2, even also in 2009, H1N1, all of them are, derived from the human influenza together with the animal influenza, reassortment. So actually, this means that the animal emergence of the the viruses may play some role in the emergent viruses. So actually, from that beginning, during the fighting against different viruses, actually the antivirus activities also pushed the progress of the sciences, like the vaccine development, drug development, and also the modern systems for the virus surveillance. So when we go back to the COVID-19, so actually we can see, this is data from yesterday, and also we can see, it is more than 400 million, confirmed cases all over the world. So actually, from the very beginning of the pandemic, actually our institute, the National Institute for Viral Disease Control and Prevention, China CDC, have done some work on the, the identification of the virus. We sequenced the first genome and also shared it with through the GCID, and also we published paper in the New England Journal. And also, actually, we developed the first testing reagent in China and shared with the local CDC to test the first batch of the patients. And also, we take the picture of the first isolated virus at early January of 2020. So in the same time, the laboratory work, actually our institute and have also focused on the in-field, the epidemic, ological, surveillance, and also the emergency, responses in different cities of China. In the very beginning, we worked in Wuhan, for the original tracing, and also go to, Northeast China to, deal with the, imported cases in the Suifeng, Shulan, Harbin, and also back to Beijing for the cold chain related, epidemic in Beijing in Xinfadi market. So actually, in the same time of the, emergency responses during the, COVID-19, because that is our job actually. And also in the same time, our lab also worked on some, the basic science like the, T-cell immunology against the different, emergent and emergent viruses. Actually, sometimes I just like to compare the immunity and the viral infection into the, this computer game like the zombies and, plants. So actually, it is quite similar. Actually, the one we face in the pathogens, actually we need the, humoral immunity and also cellular immunity to fight against the, different emergent and emergent pathogens. So actually, especially for the T-cell recognition, we need the, processing and presentation and also recognition of the T-cell epitopes, in the antigen cells, and also then, recognized by T-cells. So actually, the T-cell epitopes just, play a very central role in the T-cell recognition. And also, I also like to, compare this into the, like the Satan, like, the T-cell epitope. Sometimes it is very conserved, right? It is just like the hoofs of the Satan, and actually, people can recognize the epitope, to see, okay, it is, the virus infected cells. It is like the recognition of the Satan. So actually, in our lab, we just developed the, efficiency and also, rapid, platform for, the T-cell epitope identification through the prediction and also binding confirmation, and also, we use ELISpot, tetramer, ICS to determine, also the structure determination to confirm, whether the peptide is truly, T-cell recognized epitope. Actually, about 12 years ago, we just, identified the first, the T-cell epitope from the M protein of SARS coronavirus, and also we used the, the in vitro, T-cell in vitro MHC refolding and also the P2 cell binding assays to determine the, the binding affinity of the epitopes to the MHC molecules. And also we used the animal models to determine the in vivo functions, like, we just used the DNA vector, the vaccines to infect the, the transgenic mice and to detect the, the M protein derived epitopes. And also we detect the, the T-cell responses in the SARS recovered patients. Actually, they are recovered from SARS infection for about 20 months. And also we still can detect the T-cell epitopes. Actually, we, draw the T-cell position inside the transmembrane domain of the M protein. And also we found this is a very important antigen for the potential, the universal vaccine target for the, SARS coronavirus. But actually we can, maybe this is something, reference for the SARS-CoV-2. So actually after that, we also determined HLA A2.1, restricted T-cell epitopes from N protein of SARS coronavirus. And also we determined structure of the epitopes, and we find a really special, the immunodominant epitope presentation strategy. It is show that the bulge, let me show the pointer. So actually this is bulge, the, conformation of the peptide. It is really, higher for the, peptide, residue presentation. It is can recognized by T cells very very easily. So actually I just compare this to the dancer. Actually dancer just, stand on her tiptoe and to touch higher. So this is, kind of, the peptide to touch higher for T cell recognition. So actually after the SARS coronavirus T-cell epitope identification, we also used, the MERS, the peptide to determine any, the potential epitopes within the MERS S protein. And actually we also find some, immunodominant epitopes within the S1 and S2 protein from MERS coronavirus. And also we just determine the structure of the T cell epitope presented by the F2KD molecules. And also we found that that is, like the have a special, conformation, actually the MHC molecular have a tryptophan 73 have a, different conformation compared to other viruses. So actually it can also touch higher and maybe recognized by T cell, receptors. But this residue is from the MHC itself. So actually we determine whether this epitope can contribute to some protection to the infection. And actually we just infect the virus to the the mouth, and actually before that we just inject the peptide. Actually we can see even one single peptide can protect the mouse, not completely, but also just Okay, thank you very much. Okay, I just continue my T cell studies. Actually from the very beginning of the SARS coronavirus and also MERS coronavirus, actually we established the lot of different the virus derived T cell study platform in our lab, but actually including influenza and also the ZIKV viruses. And actually we prepared a lot of different tetramers for different emergent and re-emergent viruses. So actually I just go to the the coronavirus CoV-2 for the our studies. Actually in the very beginning of the pandemic, in 2020, actually we just actually in the very beginning of the COVID-19 pandemic, actually we just detected the the cytokine storm in the first batch of the patients. Actually we detect about 48 different cytokines within the plasma of the patients, and also actually we detect about over 38 different cytokines can increase, and also they are related with virus load. So actually after that we also collaborated with our partners in Wuhan. We just detect the single cell sequencing of the PBMC from the COVID-19 and compare it to the influenza patients. So actually we use single cell sequencing and also cfRNA sequencing together to determine the profile of the T cell responses to COVID-19 and SARS-CoV-2. So actually we used the different longitudinally collected different PBMC from the patients from both COVID-19 and influenza A infected patients. So actually both of the different patients, they shared the increasing of the the plasma cells for their cell populations. That means the robust B cell responses within the patients and produce the the antibodies. And actually for the T cells, actually they have different the activating profiles. For the SARS-CoV-2, they have the majorly it is the CD4 T cell activation and related with the different cytokine and proliferatory cytokine pathways. So actually for the influenza viruses, they have a different pathway for the the activations. So actually majorly it is for the COVID-19, it is about the the STAT1 and interleukin-6 receptor activation, but actually for the influenza it is about the STAT3 and the NF-kappa B responses, pathway responses. So actually after that we also determined the cfRNA within the plasma of the patients, and also we did not find any the virus RNA within the plasma, but actually we definitely find the T cell responses in the the plasma, and actually especially for the interleukin-6 receptor, actually it has a very higher activation level. Interestingly, actually we find that the microRNA 451A, this corresponding microRNA related to the interleukin-6 receptor, have a lower level compared to the previous, compared to the health control. And also actually, the binding site of the 451A is 3 different long non-coding RNAs. So actually, these 3 different long non-coding RNAs have a increasing level. So actually that means, actually, the upstream of the interleukin-6 receptor have been determined by our study, and this maybe can be used as therapy target for the cytokine storm. So after the determination of the patient's T cell response characterizations, actually we follow up the patients for 1 year. Actually, we determined the antibody and T cell memory within the patients, and actually we used the one cohort from the Macheng of the Hebei province, and actually in that city it is how over 200 patients in the first wave of the COVID-19 pandemic. Actually, we recruit 40% of the patients, and it is about 100 patients. And we use different method to determine the antibody and the T cell memory responses. And actually for the B cell responses, actually we determined that it is about 95% of the patients have a higher antibody, especially the neutralization antibody within the convalescents after 1 year recovery. So actually for the T cell memories, actually we also determined that it is about more than 90% of the patients have a very high T cell memory within the patients after the 1 year recovery. And actually this is, we determined the responses using the, in vitro cultured PBMC. So that means, actually we need to culture the PBMC out in vitro to determine the T cell responses. Of course, we use the tetramers to determine the specific T cells in the recovered donors and after the 1 year recovery. So actually that is for the T cell responses from the SARS, MERS, and also the SARS-CoV-2 viruses. Actually, in our study, we also determined the structures of T cell epitopes presented by MHC moleculars. And also, actually, we set up a very, the efficient platform to determine, the MHC moleculars not only from humans but also from different animals. Actually, we just use this Chinese 12 animals. Maybe everybody know this. Actually, we determine nearly all the 12 animal MHC moleculars except the snake and the dragons. So actually, we also determined the structures and published some papers as the cover stories. And especially, for the bats MHC moleculars, it is Ptal-01. Actually, this is published in biosafety, published in the PLOS Biology and commented by the Nature Immunology. And we just determined this MHC moleculars, and from one side to explain one bat can have a different immunity to the emergent and the re-emergent viruses. And actually, the bats have 5 residue of the 3 residues insertion in the the MHC moleculars. It is different from other mammals, but actually it is have a similar insertion with the lower the animals like the marsupials. And actually we determine 6 different structures of the bat MHC moleculars together with the the bat in the special viruses like the hantaviruses and the H17N10 influenza viruses, and also Ebola and the MERS coronaviruses. And actually, I will not go to details, but actually we determined the detailed structure about the 3 residue insertion in the MHC moleculars of bats, and it is have a very tight hydrogen bond network, and make sure the presentation of the T cell epitopes has a higher efficiency. And also we found that at the C terminal of the peptide, they have a special anchor residue. It is the proline, actually the proline as an anchor residue for T cells. The epitopes is not common among other mammals, but it is can be presented by the bat MH molecules. So actually also we compare this to the Ru-Yi, it is symbol of good luck in China, Chinese, the traditional cultures. Actually, this make sure the peptide can be presented by the bat MH molecules very proper. So lastly, I would like to introduce two different English journals in our institute and in China CDC to publish any relevant papers about biosafety and also about the diseases in China CDC Weekly. So actually, I would like to thank to our group in China CDC and all our collaborators these days for the contribution to our work. Thank you very much.
Fantastic. Thank you very much indeed, Professor Liu, for joining us today. That's great. Because we haven't got too much time, we're going to move your questions, if that's okay, into the next session, which is going to be the panel discussion.